Today I'm going to talk about RNA sequencing, RNA-seq. And this talk isn't going to be about a single biological system, and it's also not going to be about laboratory aspects of how to prepare your sequence and the specifications of the sequencing machines. It's going to be about bioinformatics aspects of data analysis and how basic theory, and then some examples from various collaborators that have been working with us where we've been doing data analysis. Okay? So with that, unfortunately, I have to reach over here. So RNA sequencing is a way to measure the transcriptome. It takes advantage of the rapidly dropping costs in DNA sequencing, and it measures gene expression in a true genome-wide fashion. So you can sequence all the RNA. It's not dependent on having a map of the genome and a specific set of probes that correspond to specific transcripts. Whatever's present, you can capture and sequence. It also, in addition to measuring gene expression, you can measure mutations, alternative splicing, allele-specific expression, and fusion genes. And I'm going to show some examples for most of those. Literature reports say that RNA sequencing is more accurate, and it definitely has a greater dynamic range than a microarray. It can also be used to detect small RNAs like microRNA, non-coding RNA, etc. Although these targets are dependent on how you design your experiment. This is just to show what I mean about the rapidly dropping cost of sequencing, and that the angle of drop in cost per base pair sequenced, if anything, is increasing. It's getting cheaper at an ever faster rate. And I believe it's been in the past 6 months or so that RNA sequencing has definitely been proven to be cheaper per sample than a microarray. And the real challenge of cost now is the time for data analysis and the costs of data management. So if we can streamline and automate the data management process, then we can continue to bring down the cost of RNA sequencing for a variety of different types of experiments, and perhaps improve the quality of gene expression measurements by doing more replicates, deeper sequencing, etc. This is a slide from Illumina showing that their RNA sequencing is very comparable in terms of measuring gene expression and measuring differential expression compared to quantitative PCR, which is considered the gold standard for measuring gene expression. So same sample measured, or same pair of samples measured on qPCR and RNA-seq, very high correlation. Oops. Oh, bummer. There we go. The RNA-seq that I'm going to talk about is done on the Illumina platform, mostly here at the Genome Technology Center lab at NYU. And this, if you aren't familiar with the Illumina, I'm not going to go into great detail, but in quick overview, you start with DNA. In this case, it would be total RNA, not genomic DNA. You chop it up into fragments. You attach adapters that are specific for the Illumina system. You apply that to a flow cell where there's an amplification step, which is usually called cluster generation, where each individual molecule turns into a small, oh, I forgot. Each individual molecule of template turns into a cluster of identical molecules by a series of in situ PCR. That goes on to the Illumina machine, where you get sequencing by synthesis, which is very accurate. It adds nucleotides one at a time, so there's relatively low rates of substitution and relatively very little insertion-deletion error, about 0.1% error on average. And then we get sequence data out. An important question that people ask us all the time when they're planning an RNA-seq experiment is what depth of coverage? How much sequencing should I do on my sample to get an accurate measure of gene expression? And of course, a question like that is always answered, it depends. But the facts are that you could probably get away with some multiplexing, adding several samples per lane, because the sequencers are increasing their density and output per sample very rapidly. And studies have shown that there's definitely a plateau. As you sequence more, you discover fewer new genes. And you discover, at this point, they used a cutoff called FPKM 0.05, which I will describe in a couple of minutes. But that's a pretty low cutoff, and we actually prefer to have a little bit higher threshold for calling a gene to be detected and whether we can measure differential expression. But in any case, for whatever threshold, the shape is the same. So at 100 million reads, you get 80% of the genes detected that you would detect at 900 million reads. So, running 8 more lanes of RNA-seq on the same sample will only give you about 20% more genes. Now, if your experiment is extremely focused on an extremely low expressed gene, that extra cost may be worth it to you. Most of the time, people feel like this is a reasonable bargain here in the 50 to 100 million reads per sample that acquires most of the data that you're going to get at a deeper rate of sequencing. This is a little bit about the Illumina standard RNA-seq kit. And it's important because what you get in terms of RNA sequencing measurements is quite dependent on how you prepare your samples. This is their standard protocol. You start with RNA extracted from your tissue. And you need to make cDNA in order to put it on the Illumina system. It sequences DNA. And they use a random primed kit plus a poly-A tail at the same time. So you get some bias towards the 3' end of your transcripts. They're captured more efficiently than those transcripts out here that are only captured by the random primer. After this round of random priming, then things are sheared. So you do an amplification, then a shearing. And that's important because it turns out, then you do size selection. So you amplify, shear, and size select for pieces that are between 200 and 300 bases in length. And that has an effect that it's relatively hard to capture the very ends of the transcript, because many of the fragments that contain the very end are sheared to below the minimum size. So we always get a little tapering off at the ends for a well-balanced sequencing library. It goes on the sequencer, and then we map it back onto the genome. And I'll get to that in a minute. So this is data that we actually produced from some different samples produced from different labs using relatively different sample prep methods. This is the standard Illumina sample prep method, which shows a pretty strong bias towards the 3' end of your transcripts. This is normalized to transcript length. So this is 100% of transcript length here. Or actually, this is the 3' end, 100% to the right, and this is all the way at the beginning. And so what you see is that the coverage is higher here, and it tapers off as you head towards the 5' end. And that's OK if you're comparing two samples from two different tissues that have been prepared in the exact same way, because what you're usually looking for is differential expression. However, if you're looking for something like alternative splicing or snips, variants, then your coverage is lower at the beginning of transcripts than it is at the end. And that can have a big impact. Or if you've got samples that have been prepared in different ways, then the differential coverage is going to impact your measurement of gene expression and differential expression. This is an example where the coverage was extremely even, but it tapers off at the 3' and 5, the 3' and 5' ends due to that chopping of small fragments. So you can get all three of these different distributions of coverage on the transcript depending on how the sample is prepared. Has nothing to do with the bioinformatics analysis at the end.
Sorry, so the middle one is all just random primes?
Yes. I don't know exactly how they did this. This came from an outside collaborator, but I'm saving it because it's extremely beautiful. But clearly, tinkering with your sample prep method is important. And there are numerous different kits available. Another factor, some people are interested in small RNAs, and they may be interested later on. It may not have been their specific aim one of their project, but they throw it in there as we're talking. Oh, and I'd also like to look at non-coding RNA. I'd also like to look at microRNA. I'd also like to look at SNO RNAs. It depends on how you prepare your DNA. These are two different samples of RNA that are prepared using two different kits, both sold by Illumina. And so this one has a poly-A capture first, and then it does this DSN sample prep method. This one, no poly-A capture first, and you see this SNO RNA only in the second sample. So what you do before you put the sequences onto the sequencer greatly influences what we get out. Just in general, the early stage of data analysis goes something like this. You put your sample onto the sequencer. It adds nucleotides one by one. It takes pictures. It image processes and turns those dots into intensities. Then it turns those intensities into base calls. And then it outputs the base calls as sequence reads of a length that you specify before we start the project. You want 50 base reads, or you want 75 base reads, or 100 base reads. And that, of course, influences the cost of the experiment. You can also do double-ended reads, paired end, read from both ends of a fragment, which is becoming more popular for RNA sequencing. And I'll show some examples of that in later slides. Once you've got the sequence data, then you need to go through a computational step where you match the reads back onto the genome. And that genome probably has an annotation in terms of the locations of exons and introns, which is then computationally translated into gene expression measurements. The alignment step is computationally expensive, so I wanted to put in a plug here. Oops. Coming up in another slide for our computing facility. I also wanted to point out that a lot of the quality control can only be done after you've aligned the sequences onto the genome, because that's when you know what you've got. So you can do all the quantitation and gel running you want on the sample before you run it through the system, get the sequences, map the sequences back to the genome. You won't know very much about the quality of your experiment. So we have a lot of QC steps in our data analysis pipeline. And most problems and issues from RNA-seq show up in the bioinformatics pipeline. This is what the data files look like. So that intermediate step where we've run it through the sequencer, but we haven't mapped it back to the genome yet. This is what you might get if you send your data out to a commercial sequencing facility and don't pay for additional bioinformatics consulting services. They will send you back a file like this with perhaps 200 million lines per sample. So sizable file, many gigabytes. I should also point out that for every sequence string, there's a corresponding string of quality values. And that plays into our next step, which is to look at the QC of each run. We've been using this tool called FastQC because it gives a nice graphical output, which is easily understood by the customers. So the idea is that this is a quality scale where 40 is extremely good, And anything below 30 is generally, which is, I guess, 1% error, is generally considered not acceptable. And this is the length of your sequence. This is only a 36-base sequence. We're using 50s and 75s and 100s now. But you can do gene expression with 36-base reads if you want, and probably get your data slightly more cheaply. Illumina systems tend to taper off at the end. Really good runs look like this, where the mean stays up here and the error bars don't drop very far. So this is a really excellent run, and essentially every one of these reads is going to be usable. Sometimes you see a big tapering off of quality at the end, and you can either trim them back or throw away reads that have more than a certain number of bases below a certain quality. So that would be a post-processing bioinformatics-based data filtering. This is just in general a more complicated view of the workflow. So this is the output of the sequencer. It goes through an alignment step to the genome. And then you have some sort of reference annotation information, the location of exons. And then you're going to count up the number of reads for each annotated unit, each exon, each gene, each transcription unit, other units that you might want to decorate your genome with. Then we have to do normalization, which I'm going to describe in a minute, and measure of differential expression, which is primarily a statistical issue. Once you've got a list of differentially expressed genes, then you can use all types of methods that have already been developed for microarrays to look at functional enrichment, pathway enrichment, etc. So I talked about alignment. This is the most computationally demanding part of the work. The reference genome is large. Usually we're doing experiments on human or mouse, although people have brought us RNA-seq on a variety of other organisms, some model systems and some completely novel. And so it's possible to do this on any system where you have some sort of reference genome. I'll describe a little bit later what to do if you're doing RNA-seq on an organism that has absolutely no reference data. Is this where we get the picture of it? Nope. So, so new methods have had to be developed for sequence alignment because traditional methods like BLAST and Smith-Waterman are just too slow to align 200 million reads to a 3 billion base genome in anything like a realistic amount of time. When people do an experiment, they want their data back. There's a certain amount of time that the sequencer runs, and no one can argue that they want their data back sooner than the sequencer is done with it. But once the sequencer is done, a certain amount of impatience creeps in. So we've tried to design a compute system that will allow us to do this alignment in 24 hours or less. So once the sequencer is done, we should be able to get you the first stage of data, which is aligned and gene counted, by the next day or day and a half after. I'm not going to talk about the algorithm because this isn't a lecture about that. All right, so here is the compute system that we've built with the help of Stratos and Eric there and there. And basically, this thing just has to scale as we add capacity to the sequencers. As they churn out more data per week, we need to add more CPU power to keep up with it. There isn't really an alternative. It's not like the algorithms are just going to magically get faster or the data is going to get smaller. And as we add more sequencers, we have to run this on more runs of data simultaneously. So again, scaling is important. Also, obviously, data storage is a big issue. And I'm not going to talk about the economics of that, but when you generate big data, you're going to have to pay for some amount of data storage over time. And it's not necessarily a good idea to download it onto a portable USB drive and store it on the shelf in your lab. Having a backed up system where all the runs are archived in some sort of organized fashion is probably going to be necessary for your research. We had the system set up so that the sequencer writes the data directly onto our hard drive array, and then we have direct access to it to run the data analysis algorithms, and then you in the lab, wherever you may be, can get direct access to the raw data and our analysis results.
What's the range of data, amount of data?
From a few gigabytes to many hundreds of gigabytes per experiment, or per yeah.
Was that before or after the analysis?
Yeah, it grows as we do more analyses, right? And so frequently you need to do it this way, and then that way, and then the other way, and those things just grow.
And so by the time you're done, you're in the terabyte range? You can be.
Can be. And also it depends how many samples were in the experiment, et cetera. It can be, but usually hundreds of gigabytes is a typical project. This is just an example of the raw output that you would receive if you ran a sample through the sequencer. This is an older one, but the clusters represent those little spots of light that I showed you on the first slide. Some of them don't make it. They're not bright enough, or they overlap with another one, and they're filtered out at this point. This is called clusters pass filter. So this number is always a little smaller than that one. And it tells you what % of those clusters pass the filter. This is basically not your problem as an investigator. It means how accurately the machine was loaded, because it has an optimal concentration of reads per lane that it can produce. If you underload, then your pass filter cluster % is high, but your total number of clusters is lower than it could be. If you overload, then the spots start to sit on top of each other, and you get more errors during the sequencing process. And so you get a higher number of clusters, but a smaller passed filter %. And the techs in the lab are constantly adjusting their reagents and various parameters on the machine to try and optimize this, to get the most possible data. Because it's OK to lose some if on whole you get more. Also, we calculate an alignment %. That means how many of the reads that you produce match the reference genome of the organism you're interested in. In an RNA-seq situation, we also have to deal with gapped alignments, matches that contain introns. You've sequenced the cDNA, now it aligns, but in 2 parts. And that's a challenge for the alignment software. And several different software packages approach this in different ways, which I'll talk about later. You never get 100% for several reasons. First of all, there's some amount of contamination, which represents, in this case, ribosomal RNA. Now, we do a poly-A selection, so we get rid of a lot of that, but some creeps in. Mitochondrial RNA also creeps in. DNA from other organisms that might be contaminating your sample. We also can't align highly repetitive sequences. Stuff like alus, centromeres, telomeres, they align in many, many different locations all over the genome, and the algorithm just throws them away. Typically, for RNA-seq, you don't get a lot of centromeres and telomeres, and something in the neighborhood of 80 to 85% is quite good. Anything less than 70%, we get out the QC tools and start looking at what happened to your sample. What is in this batch of sequences that fail to align? Are they all ribosomal, so your poly-A wasn't very efficient? Is it a virus contaminating your sample? That happened to us once. Is it some unknown DNA from some other known organism? That happened to us a few times. Have you made a cloning mistake and sequenced the vector a few 100 million times? That happened to us more than once. This is a summary file from very recently. We're now getting close to, well, between 150 and 200 million raw clusters. Range here. And the range, again, has to do with how precisely we're able to quantitate the DNA, mostly. Various substances in your DNA prep may influence the quantitation in various ways. Again, our alignment percentages are mostly in the 80s, and our alignment scores are good. Now we also get a new measure called the percent greater than Q30. Remember I showed you that pretty colored graph that slopes down? They've put a floor on it at Q30 and said everything above Q30 is usable, everything below is not. And they're saying, well, we're getting 95 to 96% of all sequence data above Q30. So this is a really good read, which is why I show it to you. OK, so the challenges of alignment that I mentioned, there's a problem of mapping to a reference genome because of the introns. Small reads may have many short overlap at one end of the intron or the other. So you could imagine a gapped alignment tool, but there's some limit to how smart that tool can be. If you've got 2 or 3 bases that span the intron on the far side, it's not going to be able to find a perfect match for those 2 or 3 bases. So what do? Do you just chop it and live with the shorter alignment and call it a good alignment? What's the threshold for that? What if 10 bases don't match, 15, 20, etc.? So what to do with reads that map to the introns or outside of intron boundaries? Also, the annotation of the genome allows for some overlapping genes. Which gene do you call when you've got a read that overlaps 2 different ones? And there's a variety of different algorithms for this.
So you always make the same size of reads?
Yes. On an Illumina system, all the reads from one experiment are the same size. You can choose, actually, it's like a menu. You can choose 36 base reads, 50 base reads, 100 base reads. We sometimes get 75s, but not in the current batch of kits. There's a little kit that has a whole bunch of reagents, and that actually determines how many you're going to get. And how you choose that, there's, again, a whole lot of factors that come into play. Longer reads, you're going to get across some small repeats and get a slightly higher alignment percentage. You're also going to be able to align on both sides of introns a little more efficiently. It really depends on what your goal is. For a lot of pure expression studies, the shortest reads are perfectly fine. 36 bases is enough to map over 80% of your fragments correctly to an exon. I'm not going to go into great detail, but there's 2 popular methods at the moment for doing this alignment. Illumina has its own software called Cassava, where they rely entirely on a reference. So you go into the reference genome, say RefSeq for human, and you just take all the exons that are annotated on the genome, and you find all the splice positions, the end of one exon and the beginning of the next. And you fuse them together. So you have what they call a junction read. So if you were on a cDNA, you'd be running off one exon and then right into the next one. And they take a junction read that's a base shorter than the length of your read. So if you have a 36 base read, they take 35 bases from either side of 2 exons, and they put that into a separate data file. Now when you go and do the mapping, you do it in 2 phases. First, you map all the reads to the genome. That gets the ones that fall entirely inside of exons. And then you map those reads that are left over to your library of junction fragments. And that catches usually another 20% of your reads that correctly span the exons. And in this case, it doesn't matter if it only spans the exon by a couple of bases, because it hits the junction fragment perfectly. A completely different approach to this, called Top Hat, uses an algorithm that allows for gaps. It says, OK, we know that this is a gappy alignment process. So when we have a minimum alignment size of, I guess it's 24 bases, and then it stops aligning, we know that we've run into an intron. Now we take the rest of the sequence from the point where it stops aligning, and we move down the genome looking for a region that looks like a splice site. And we test each splice site to say, OK, does it align here? Does it align here? Does it align here? When we find a spot that the rest of the sequence aligns, that's saved into memory. Other reads that are in the same genomic position are also tested. And a splice site becomes real when several reads match across that splice position. I got a graphic about that.
How much smarts goes into the selection of possible sites? Is it just?
It's a pretty simple pattern. It's 2 letters.
Okay. All right.
But it uses those 2 letters as a seed, and then it tries to build an alignment at that position. So it doesn't have to test every possible alignment, and it also has a maximum intron size, 100,000 bases or something you can choose. You could set that if you want.
Broad. It's pretty.
Yeah, and they've actually generalized this to another algorithm that I'm going to get to in a minute, which is really interesting. A third way of doing this, if you had no reference at all, is to take all of the reads and do de novo assembly. Just match all the reads to each other and try and build complete transcripts. As you might imagine, this is very computationally intense for 200 million reads. And we don't do it very much. But if you need us to, we can. This is just to say, in general, this method works pretty well. And once we're done and we look at the map of the reads on the genome, we see that they line up really well with the annotated exons. Of course, if you think about it, it's sort of backwards reasoning. We use the exons to map the reads, and then we show that the reads map onto the exons. So all the ones that don't map mostly go away. But we always see a few reads that map onto intron positions. So this is a very typical look of an RNA-seq project. This one is also showing you differential expression of this gene between 2 samples, which I will get to in a minute. And I have to credit this data comes from the Costa lab.
So Stuart, the cassava can address cases like this where you have match into intron?
Yes. The first step of cassava is take every read and map it to the whole genome. So it only fails where there's gaps, which is presumably an intron. So it really fails completely where there's an intron that's not predicted. So if you have a read here that's split with an upstream exon that's not annotated, then you're going to miss that. You may see a peak for reads that map entirely in that upstream exon if it's big enough. If it's small, if it's 22 bases, then it'll be missed entirely. So this is an example of that algorithm where it's looking ahead, trying to find where the second half of the read maps. It looks for an AG site and says, OK, does my read map here? And then can we put, after we build a putative junction fragment from here to here, do more reads align? So this is from the Top Hat paper, Trapnell et al. Bioinformatics. So if you're interested, go read up on it. It's a pretty cool algorithm. And it's been the basis, is sort of like the foundation for a whole pyramid of additional software. OK, so this is the stuff that we need to do. Filter out the ribosomal and other sort of junk that shows up in your sequence. We can build a junk filter file so that in addition to mapping your sequence reads to the genome, we can also map them to the junk file. That way we know right away how much ribosomal RNA was in the sample, how much tRNA, et cetera. That's relatively straightforward. Align to the genome, find the splice junction, count up the reads per gene, look at differential expression. We can also try to look for alternately spliced transcripts, novel genes and exons, SNPs, allele-specific expression. I'm going to get to most of these. All right, so differential expression is, in theory, very straightforward. You're looking for cases where in one sample you have lots of reads and in another sample less. The challenge is usually about normalization. Every lane of the machine produces a different number of reads. I showed you several examples of that. It depends on how you titrate your samples and whether the solution is cloudy or whatever. So you're going to get somewhat different numbers. If you just count reads and compare, the counts are going to be influenced by the total number of reads that were collected. Just like in a microarray, the brightness of your hybridization is going to be different for every array that you run through the signal processing. So we have the same problem. We need to normalize. Simple way to normalize is to divide by the number of reads in the sample, or per million, in other words. So that's just a scaling problem. Divide counts per million reads. Another related problem is that a longer transcript will have more room for reads. It will produce more fragments in the library and therefore will be sequenced at a higher level than a very small transcript for an equivalent amount of gene expression. Usually you think about gene expression as transcripts per cell, not in terms of base pairs of RNA per cell. And so you want to normalize the number of reads per gene to the length of the gene. Pretty simple. And this was proposed as a method called RPKM from the Wold lab at Caltech in one of the very first papers on RNA-seq. And I forgot the reference here, sorry. It's Mortazavi. And it's called RPKM, reads per KB of transcript per million reads in your sample file. And that works reasonably well as a normalization. There's problems with it that I'll get to in a sec, but in a number of comparative studies, RPKM works okay. So if you RPKM normalize a bunch of samples and then look at differential expression as simple fold change, you can get results. They may not be the most statistically accurate, but they are results. The RPKM makes a few assumptions. It assumes that the total amount of RNA in the cell is constant between your two samples that you're making a differential expression and calculation. Microarrays make pretty much the same assumption, that the total signal on the array is constant because it comes from all the RNA that comes from your tissue. And then it also assumes a baseline that most genes don't change expression, a few do, so that you can normalize based on some sort of mean weighted average. And that's not such a good assumption. There's a lot of biological experiments where most genes are making big changes in expression. We have some heat shock experiments where most genes are hugely downregulated and a few genes are hugely upregulated. And then the RPKM assumptions are violated. How that affects your data? Well, the genes that change expression a lot are going to squash the genes that don't change expression much, and you're going to bias your calculations. There's also the situation where just a few genes have a very dramatic change. A few high-expressing genes have a very dramatic change. And the problem here is that, unlike microarrays, RNA-seq is a zero-sum game. You have 2 million or 200 million reads, and they're distributed among all the different genes. When one gene is using up a lot of those reads, all the others are squashed down. That doesn't happen in a microarray. Each hybridization chamber or cell, or whatever you want to call it, is more or less independent in measuring its signal. Whereas here, the signals are interdependent. A few things using a lot of RNA are going to reduce the signal from everything else. And so normalization methods are trying to take this into account. But if you design a normalization method to explicitly take this into account, you're probably creating other problems in samples where that's not happening. So it's a real challenge right now, and there's a lot of different papers being published in the statistical literature, and there's not a perfect solution. Probably for very different types of experiments, there are different optimal statistical methods. And there's probably not one statistical method that's optimal for all types of experiments. The methods that are popular right now are using quantile normalization. And we've definitely tested this fact. Different published normalization and differential expression methods give different results from the same data. And people aren't thrilled about that. The best we can do right now is use several different methods and give you the intersection and say, OK, this gene is showing as highly differentially expressed by several different methods. It's at the top of the heap. And we could even calculate a rank across several different methods and say the average rank of this gene in differential expression across several methods is as follows. And that's a more stable measure. It's a data fusion method. I said this already. Okay, just in general, when you're looking to measure differential expression, that you're measuring a rather variable quantity, gene expression. And if you measure gene expression from several different samples that are biologically identical, there's going to be some variance in that measurement. Everyone knows this well. RNA is labile, and there's a lot of things that influence the expression of tens of thousands of different transcripts. So in order to say whether a particular biological treatment has changed gene expression, you need to have some measure of the variability in an unchanging situation, which means that you need replicates, and you need to measure the experimental variability in your system. Unfortunately, RNA-seq samples cost money. And in order to get an adequate number of replicates, in order to have statistical power to measure the variability and set your error bars appropriately, you have to do more replicates. This is where we go back to my earlier slide where we showed the cost of sequencing going down and the number of sequences that you need in order to find a detectable level for your message is finite. You don't have to sequence infinitely deep in order to get reasonable data on most of the messages. So my point here is that more replicates will give you more precision in the measurements of gene expression changes for many of the genes, except for those that are extremely low expressed, at which point you need to trade off deeper sequencing versus more replicates. But I'd rather be more sure of the results that I published and be willing to miss a few genes at the low end of the spectrum rather than have fewer replicates and a poor estimate of variability on all the genes that we measure. Yeah.
In terms of estimating the messenger levels, the transcriptome of genes that we know of between the two systems, the new one, deep sequencing, and the previous microarrays, that maybe suffer less from all these normalization issues.
Well,
what do you recommend for a lab that's just interested in
a few genes
into the genome, the transcriptome? I mean, not the full transcript, just the genes.
Which genes? Just a few genes or all of them?
Coding genes.
Coding genes. So you're talking 20 to 50,000 different transcripts, right?
Right. Everything that's covered by the normal Affymetrics, the typical Affymetrics.
Well, what we see is good correlations between the two. I can say that, regardless of the details of your statistical methods, even if you just do the simplest, the RPKM and the t-test with multiple test correction, you're going to get good correlation between microarray and RNA-seq. RNA-seq is increasing in popularity because it's becoming cheaper, and you can reuse the data for other purposes. I don't think it's better than microarray, although it certainly has a better dynamic range. So you can see genes that are expressed at a lower level here. You get better accuracy for genes that are expressed at a really high level. And the prices are coming down. I mean, you can do one experiment and compare.
In your first plot, you had a very impressive correlation. Do you know how they did the calculation for that?
Illumina internal data. They did it so it came out good, I think, is their answer. And they also did it on a finite number of genes because they were qPCRing, so they probably did like 500 genes. We usually set our threshold at RPKM of 1. So we ignore all the genes that are expressed at a level of less than 1 gene per million, 1 transcript per million. We might push that up a little now that we're getting 200 million reads per sample, unless we start to multiplex and we bring the number of reads per sample down to 100 or 50 million. This number is supposed to be equivalent to 1 transcript per cell, in theory. So, you know, your tissue, you've got millions of cells, but things that are lower than 1 transcript per cell are probably not having a lot of important effects, unless your system is an unusual one. Okay. So this is just an example to show that we can get this done. Some genes show strong differential expression, and it probably doesn't matter what statistical technique you use. They're going to come up near the top of the list. They're not, this effect is not going to disappear with a different normalization method. When we rank our genes by differential expression or by p-value, and then send that data back to the lab, they pick their 20 or 50 or 100 favorite genes that are near the top of the list, and they're able to validate about 70% of those as being differentially expressed by qPCR in the Marullo lab. But this holds up for most of the other labs that we've worked with. I've heard 70, 80, 90% of genes that have high differential expression scores from our statistical analysis are also validated by qPCR. Same as microarrays. 70, 80% validation is pretty good. I'm going to talk really briefly about this, although lots of people bring us alternative splicing problems or questions. The theory is that genes are spliced differently. Certain exons are used sometimes and not other times. There's upstream and downstream exons. There are internal positions internal to the exon or internal to the intron. That's where splicing can occur. The net result is that there's a lot of different transcripts in your total RNA isolation mix. To some extent, we can differentiate these. Certainly in the case of this purple exon, or in a read that skips it, we could tell the difference based on the number of reads on this exon. We can say, yes, this exon is expressed at twice the level of this one. Alternative splicing exists in this system. We see at least 2 or maybe 3 or 5 different transcript types. That's relatively straightforward. A different question is, does the alternative splicing change in my biological system when I do an experiment? Harder question to ask, particularly if you don't know what gene you're looking for. If you do know what gene you're looking for, then it's a relatively simple question to ask. Does the relative expression of this exon change with respect to that one? And we could give you that with good precision. On the other hand, if you want to look at the entire genome for every exon and see if it changes with respect to any other exon in the same gene, that's a lot of comparisons. It's in the hundreds of millions of comparisons, and it gives you a very, very high multiple testing correction and a very, very high chance to find false positives. This is even more problematic when I show you that coverage curve where there's 3' bias. Those areas of the transcript that are covered poorly are also going to be covered erratically from one sample to another, and so you're going to find a transcript shows up in one sample and not in another, and that's going to be called as differential expression by any reasonable algorithm, but it's not reproducible. So this is causing us a lot of struggle, and it's not my favorite experimental design. Find me any exon that changes expression in my sample. All right, I'm going to skip this. Related, but not exactly the same, is this idea of finding new exons and re-annotating a genome experimentally from the RNA-seq data. And this is really interesting because whenever you look at RNA-seq data, you can always see some stuff that doesn't agree with the genome annotation that we're using, especially if we use a really conservative one like RefSeq. There's always extra exons. There's always expression up here that wasn't previously included in the 5' end of the gene. And we could use this kind of a method. This is, again, the Top Hat software to find new exons and new transcripts. And so, again, if you're interested in a finite number of genes, we could do this. We can do it with reasonable certainty genome-wide. You can see some cases that make a pretty strong argument for alternative splicing, where a particular region shows up in one sample but not another. Maybe here, certainly here, maybe here. Just as an example. Costa Lab again. And I've forgotten what this one shows. Oh well, it's pretty. Actually, I think this is just an example of alternative transcripts that are annotated, and our data support that pretty clearly. So here's one form of transcript. Clearly, most of the transcripts show an exon here, but a few show a read-through, and we see that read-through in the data. And it looks like it's a small percent of all the reads. So I think this is just showing that often alternative splicing does support the annotation. OK, you can find sequence variants in RNA-seq. This is often touted as one of the advantages of doing this type of experiment, that you can come back and reanalyze your data and look for sequence variants. You could also find allele-specific expression. If you've got two different alleles, you could probably look at those transcripts and see which allele are they coming from, or even what ratio of alleles are we seeing in the RNA data. However, there's a big however here. The coverage is uneven. If you think about gene expression, obviously different genes are expressed at different levels. You're not going to get a nice even coverage the way you would with a whole genome sequence or an exome-based sequencing. So it's harder to detect SNPs in genes that are expressed at lower levels, obviously. Also, the coverage across transcripts isn't perfect. We often see 3' bias. There's high levels of PCR duplication. In that process that I showed you from Illumina, where they start with random primers, they do a couple of PCR steps in there. They have to because they need to make cDNA. And therefore, a mutation that creeps in as a PCR error and is then amplified will show up a lot of times in your reads. I'll show you that. And then there's the possibility of RNA editing, which we don't really have a good handle on. But clearly, there are sequence changes in RNA that don't reflect changes in the original DNA. And we do see that. OK. So, back. Here's an example of extreme bad data. Sorry, Jerry. I don't know why there's so many mutations in this data, and they're sort of all over the place. But it's probably not true that all of these variants are present in the original genome from which this DNA was transcribed. It would be really a nightmare to try and call SNPs from this data set, which he asked us to do. And we got hundreds of thousands of SNPs, and they didn't validate well at all. So here's an example of duplication. I'll show you more of that. So here's an example of a PCR duplication. There was one template, one read, that had these two variants in it. And then it was sequenced a whole bunch of, it was PCR'd a whole bunch of times, and it ended up becoming a bunch of different templates on the sequencing surface. We know they're all the same because they have exactly the same start and stop position. If this was real sequence data, it would be randomly fragmented, and all these different reads would show the same mutations, whereas we do have a few overlapping reads that start at different places that don't have these two mutations. So this is a perfect example of a PCR duplicate. I've asked myself, why can't we just eliminate all the duplicates from the RNA data? Well, there's a couple of problems with that. Problem one is you can't do that if you're also measuring gene expression. I mean, you could do it in a separate pipeline on the same data set. But the other problem is you go very deep in RNA-seq, so you have really good coverage. And if you eliminate all duplicates, then you put an absolute ceiling on your coverage of 100, or of twice the length of your read. You can have exactly one set of reads overlapping each other going in the forward direction, and exactly one set of reads overlapping by one base in the reverse direction. So if you eliminate duplicates, then you put a ceiling on the number of reads. We could still do that. That would reduce this problem somewhat. But you should at least know that the problem exists.
By the way, this you get after you filtering out how many mismatches?
2 mismatches, a lot, maximum of 2.
So there, if you
But
have,
2 mismatches in the 32 base window.
Right, so this is gathered from more than 36 in the left, because you have 3, sometimes 3 mutation.
Yeah, well, what happens is if the seed matches, if you get less than 2 mismatches in the first 32 bases, then the rest of the alignment is tacked on, even with more mismatches. So if the mismatches are scattered throughout and you have no chunk that has fewer than 2 mismatches, then the read gets thrown away. But if there's a chunk that matches and the mismatches are at the tail end, which you see often because the quality tapers off, which is what you're seeing here. But again, these may be some sort of duplicates as well. Here's one that we think is real, a SNP that was detected by RNA-seq. The reason we think it's real, it's showing up in many reads that start in different positions. And it's showing up in good quality reads that don't have other mismatches.
You expect them to be 50-50 when we describe SNPs?
50-50 mean directionally?
No, owing to the expression from 2 chromosomes. So SNP.
Yeah, 50% allele frequency is a high likelihood. Yeah, so you don't have to be in all because that would be a homozygous mutation. So yeah, so your cutoff to be interesting is something that's statistically close to 50%. You might set your cutoff at 30% as being entirely reasonable. Now, of course, if there's differential allele expression, 20% might be real, 10% might be real. It depends on how you set up your software. Those are parameters that you can play with depending on your expectations. OK, so this is from Zhujian Tang again. Actually, almost all the figures are from Zhujian Tang. But this was her idea to show all the SNPs, the ones that have good confidence, and then the ones that have good confidence that are non-synonymous mutations that, in other words, might be functional to the disease that we were looking for in the first place. So just that's what you're going to expect. There's a lot of data, and it narrows down pretty quickly if you set your confidence thresholds pretty high, and you're only looking for mutations that are actually functional. So the good thing is, you could pick 20 or 50 of these and test them somehow and get a reasonable idea. If you had 100,000, then you wouldn't feel confident picking a few and testing them because you know most of them are false positives.
It's really complemented in the study with genome survey.
Like exome type sequencing?
Yeah, all DNA.
Yeah, we've seen the same pattern, with the exome data as well. It's just a matter of setting your thresholds here for what you call a good confidence. OK, a couple more slides, and then I'm going to finish. I'm not going to run till 1. So what do you do about novel genomes, something where there's not much reference data? There's no annotated exon map or whatever. You got to have some sort of reference, or else you're just left with de novo assembly. We can use predicted gene models, like based on ESTs or BlastX, any protein that matches the genome anywhere, that could be used as a predicted exon. Or you could do de novo assembly. So here's some data from the Desplan lab on a wasp, where we had only EST-type predicted genome annotation, which is very rudimentary. And as you can see, the RNA aligns to these genome fragments in places that's not expected as exons from this existing map. So we're starting a project to re-annotate the genome based on the real gene expression that we see in the RNA samples. So these are exons, exon, 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 intron, exon, et cetera. So, and then you could also use this same map to, say, do differential expression between different tissues of the same organism, or any kind of experimental treatment that you would want. So you're not completely without any recourse if you lack a good genome annotation. It could be built by bootstrapping from the ground up. If you don't even have contigs of genome sequence against which to map, then you're left with de novo assembly. And we've been using this tool called Trinity. And the main point here is that this is a very, very computationally intense process. And to run this on a 200 million data set took us a week. So it's not something that we can do on lots of samples all at once. And it's kind of a last recourse. And in fact, the transcripts that this builds are not very reliable because it's looking at this overlapping, and there's nothing to prevent it from assembling different into one transcript fragments that are actually all parts of different alternatively spliced transcripts, or vice versa, creating alternatively spliced fragments from something that really is one sequence but has a couple of base pairs mutated so they don't assemble well. So it'll give you something. It'll give you a set of transcripts. It'll definitely give you the baseline that you could then use to, say, translate into proteins and get information about the genome of your organism, but it's not very reliable. But in their paper, it looks really good. So they get really good reads on this predicted gene. OK, last major topic that I wanted to hit on, because we've had success with this in the last couple of months, and it's really exciting, is this idea of fusion genes. Fusion genes are really important in cancer. They are dominant mutations. So when a chromosome breaks and reforms at a position where a transcript exists, you could create a brand new transcript with a new gene function, which could be an oncogene. So we're looking in all kinds of cancer samples at RNA-seq to try and predict fusion genes. Oops. So we're using this software called Top Hat Fusion, which is an extrapolation of the Top Hat software I showed you before that predicts novel splice sites. In this case, it does the same thing. It does an alignment, looks for a place where your read stops matching the genome. And then it jumps, but it jumps not just down the genome looking for the next splice junction, but it jumps everywhere else on the genome. And it tries to find a place where the rest of your read maps. In order to do that, the read needs to be pretty long. It needs to be at least 50 bases, because they have a 25 base chunk that has to map, and then another 25 bases has to map somewhere else. 75 works better. 100 is even better. Also, it works really well, or it supplements its quality when you have a paired end read. When you read each fragment of RNA, or actually cDNA, from both ends. And then you map those two ends, and one maps on one chromosome and one maps on another. Those two things together, where you have a junction fragment, one read that goes across the splice site, and some paired ends that map on either side, one chromosome and then the other, is the essence of Top Hat Fusion. It also has a lot of filtering. This is where we were spending a lot of time until their paper came out, because we were developing our own method. And we found that, well, wherever there's a repeat in the genome, you're going to get some reads that map on one chromosome and then on the other. So you have to eliminate all sequences that are repeated in the genome before you start looking for these fusions. It's a real problem if your fusions are taking place in repeated genes, like TCR locus, where there's many repeats. And that's actually a place where fusions happen. So we haven't quite nailed that one. This is a graphic of how their thing works. So ideally, your read is long enough that you can break it into several pieces. One piece maps here on this chromosome. Another piece maps here on this chromosome. When you try and build a fusion, you discover the exact junction is here in this center piece. And so when you create a putative junction fragment, you can do an alignment of your read across that junction fragment. Now that you have a junction fragment, you can go back to your library of sequence reads and see if any other reads map to this junction fragment, even though maybe they're not long enough to give you 25 bases that land on the other side. The result looks like this. So you've got a combination of pairs that map on either side, chromosome 20, chromosome 17. Those are reads that come in from the ends of the same fragment. And these split reads, where one read actually can be split and reads right across the junction. This is the Top Hat Fusion paper, genome biology. And this is our data from the Roth lab, where we ran this on some RNA from mice that were known to have tumors that were induced for fusion genes. They're set up to produce fusion genes. And we were able to find some and validate them by PCR. So we're really excited about this, and we're running as many RNA samples as we can through this algorithm right now. 2 slides more about integrative projects. You can combine RNA-seq with other methods. We're mostly interested in combining them with other sequencing-based methods, but certainly they could be combined with all kinds of other biology. We did some experiments where we combined RNA-seq and ChIP-seq. We were looking at epigenetic changes that would affect gene expression, and we found some pretty good results. And we can also combine that fusion gene detection with gene expression. So this is an example where the ChIP-seq shows that there's additional methylation in the upstream region of this gene. This is the ADM gene. It's transcribed this way, and further upstream we see more methylation in one sample than in another, and in that sample there is greater gene expression. Pretty straightforward. And then we published this in PLOS ONE with Costa's lab. And this is an example of gene expression changes at a fusion site. So if you think about it, a gene fusion moves the bottom half of one gene to be attached to the top half of another gene, and it's driven by that gene, the first gene's promoter. But when you look at the alignment on the normal genome, what you see is the second half of the gene is expressed at a higher level than the first half. And it's inexplicable unless you know that all of these reads are actually coming from the fusion gene. So it's hard to find this by algorithm, but to use this to validate the fusion gene prediction is really exciting.
Do we know of any normal events that happen? Just,
to do this?
Out of curiosity.
I don't know of any.
Without any relation to cancer. I mean.
Fusions that are not related to cancer, sure.
Yeah?
Yeah, they're called translocations. They happen at some frequency in every genome.
But this would be in the...
These would be somatic translocations.
Yeah. Here you presented something which is on the level of transcript.
Yeah.
These two means...
But you would go back and validate this on the genome of the tumor cells that these transcripts came from to see that you actually had a genomic fragment that spanned those 2 chromosomes. And my thanks. So I especially want to thank Konstantin Alifaris, who's the director of our Center for Health Informatics and Bioinformatics, and Zuzhan Tang, who does most of the hard work in all of the slides that I just showed you, processing the raw data and doing the downstream informatics. The rest of our sequencing informatics group, Office of Collaborative Science that provides our funding, CTSI that provides our funding, the Genome Technology Center that does all the hard work of generating the data, Jiri, Baron, Elisa, and Adam, and everybody who gave me data, both the slides I show here and all the other projects that we're working with. Thank you. I guess I can do questions, although we had a nice round of questions during. Tom.
In your example of raw data, I think you showed the reads are 152 bases long. Was that another technology?
That was a MySeq. So we have several different machines in the sequencing lab. The GA2X gives us 36 base reads. The HiSeq gives 50 or 100s. I think it's going to go to 150s really soon now. And then the MySeq goes from 100 up to 250. But many fewer reads per sample, about 6 million instead of 200 million, and cheaper. And these things change all the time. You can also sequence using 454 technology that can give you up to 1,000 bases, much more expensive.
Is it true that the MySeq is lower in the amount of reads?
Yes, 6 million reads versus 200 million per sample. So it's typically used in different experimental designs. It's not usually used for RNA-seq. Thanks.
How do you detect tissue, allele-specific expression?
Well, you need to know the different sequences of the two alleles. And then you can just line up the RNAs and look at those bases that are different and count. So you can't do it genome-wide. You have to know what you're looking for. You have to, or, I mean, you could do it in 2 cycles. You could call heterozygotes and then go back and look at the relative levels of expression of those 2 genes across different samples. But it's not, it doesn't come out at you right away. You have to go into the data and know what you're looking for.
How are the random primers selected? Are they pulled from the genome of the organism you're looking at?
Random.
What's the length?
I don't know. Adam, do you know? I don't know. It's a kit. I mean, you could look it up on the Illumina website as easily as I could. I'm guessing 8 mers.
Oh, they're tiny. Okay.
Well, that's the idea, is they're supposed to land all over the place and amplify promiscuously. Okay, thank you.